Hi, welcome to Breeze Science Lab. Today we're starting a new system, the respiratory system. So this will just be a very general, basic introduction to the anatomy and histology of the respiratory system. We're going to start off with the definition of pulmonology, though. That's the branch of medicine that focuses on the respiratory system. So that's the lungs, airways, and all the associated structures like your vocal cords. Okay. Now, a pulmonologist specifically would focus on diagnoses such as COPD, pulmonary fibrosis, lung cancer, pneumonia, sleep-related breathing disorders such as obstructive sleep apnea. We hear about that a lot. That's something's blocking your trachea, which means it's blocking airflow during your sleep, and that causes the individual to wake up. And we don't want that. And so a pulmonologist will also study that. Now, this is closely integrated with the cardiovascular system. Why is that? Well, the cardiovascular system, the main job of the heart is to pump blood, right? But why do we need blood? Why do all of our tissues and organs need it? Well, the blood is carrying oxygen, and what we really need is oxygen. So, oxygen is important because you need it for aerobic metabolism. Anaerobic really doesn't last that long, but aerobic metabolism is really how we get energy. So think back to your MLE classes where you went over the electron transport chain and those other cycles. They all really use oxygen for energy. So our brain, our tissues, everything needs oxygen in our body. And the respiratory system is responsible for getting that oxygen. The cardiovascular system is responsible for distributing it. Now, we kind of already mentioned the main function of the respiratory system is respiration, which is gas exchange. What gases specifically? Well, we want O2 and we really don't want carbon dioxide or CO2. Also, air conduction. How does the air get to those respiration zones? Well, they have to be transported there. So, conduction is also part of the respiratory system's function. Air filtration also. So, if you think the air we breathe in, it's not just pure oxygen. You have other gases, you have debris, you have pathogens, allergens like pollen. All right. And our lungs don't want that. They have their main job and that's just what they want to focus on. They don't want these allergens getting in the way. So we have to filtrate them out. Vocalization, that's speech production. Your vocal cords are involved in the respiratory system as well. Your sense of smell, so olfactory epithelium is in the top of your nasal cavity, and that's responsible for your sense of smell or even your sense of taste because they're very closely related, right? If you lose your sense of smell, you lose your sense of taste, usually. Also, regulation inhaled antigens, that's kind of like filtration, but it's more, think immune system based. So, fighting off those pathogens if they happen to get that far. Maintaining blood pH balance. This is a really important one because we have two systems that maintain our blood pH at around 7.4, our kidneys and our respiratory system. Now, we as humans, we like to keep our blood pH in a very narrow window. We don't like 7.5, that's alkalosis. We don't like 7.3, that's acidosis. So these two systems work together to regulate that pH if it so happens to change. Now, kidneys, that takes a lot longer, right? That's going to take days to weeks to regulate. But your respiratory system can work a lot faster. You can increase your breathing rate, you can increase your breathing depth. So these will work way faster. So, for example, you decide to climb Mount Everest tomorrow. You've never been at that altitude before, but you're going to climb it anyways. You get up there, and you know how they say the air is thinner? Well, there's less oxygen up there, right? So, that's a problem. Your respiratory system, though, is going to help maintain your blood pH even though there's less oxygen, right? Because these gases are huge in maintaining that pH, right? If you have too much carbon dioxide, you have too much acidity in your blood, which causes the pH to go down. And the opposite also occurs. So the respiration system works more acutely at maintaining that. Anatomy. So, our respiratory system is actually broken down into two divisions. We have our conduction system and our respiration system. Now, respiration is mostly focused on gas exchange, right? So, once you hit that system, that's where respiration takes place. Conduction is, again, just that transport to those respiration zones. So, that's going to include nasal cavities, your pharynx, your larynx, trachea, bronchi, bronchioles, terminal bronchioles, right? And that goes in order. Now, once you hit respiratory bronchioles, that's the first part of respiration system. And then it goes to alveolar ducts, alveolar sacs, and alveoli. Now, alveoli, think about them as like little clusters of grapes. So you have like the little bunches right here, these are all alveoli, right? And those are the main part of respiration. That's the main part, but technically all of these bullet points in that column can be responsible for that as well. Lungs. You got two of them, usually, and they're covered by a thin layer of connective tissue called the visceral pleura and the mesothelium. Also continuous with the parietal pleura, which lines the pleural cavity. So what are these linings, right? Well, in your body, when you're breathing and just other natural, bodily functions, they're rubbing against each other, right? Different organs with cavities, everything like that. That's not good. Frictions, we don't like friction, right? Because friction is going to lead to inflammation and that leads to other problems. So we have these linings that glide past each other, and there's just a little bit of like fluid or air, depending on the type of pleural cavity we're talking about. But if there's too little, we get friction. If there's too much, then we have another problem because too much equals pressure, and that can constrict the lungs in this case, right? So if it gets filled with air or if it gets filled with fluid like pus or even just edema, it can constrict the lungs. Sinuses. When you go to the doctor and you're there for upper respiratory infection, you tell them, "Oh, I've just been feeling so awful. I've had a cough, a runny nose." What do? They press on your face, right? They ask you, "Are you tender here? Are you tender here?" They're pressing on your sinuses. So you have four sinuses that are associated with each nasal cavity. You have paraethmoid, paramaxillary, and parafrontal, and then you have a combined sphenoid one. And normally these have small little drainage cavities to drain all of the mucus, right? If you have an upper respiratory infection, though, it can become occluded with the mucus. And then mucus just builds up in those sinuses, eventually causing increased pressure and tenderness or even headaches, right? So that's not good. And then it can even lead to a bacterial sinusitis, a secondary one, which causes even more problems down the road, like pneumonia. They have pseudostratified ciliated columnar epithelium with goblet cells. Goblet cells are just responsible for that mucus, and the ciliated columnar epithelium is just the regular respiratory epithelium. Histology. So, what other epitheliums do we have? Well, we have olfactory in the upper one third of the nasal cavity, which is the olfactory region. So up here, that's responsible for your sense of smell. But then once you get to the lower two thirds of the nasal cavity, it's lined with respiratory mucosa, and that's generally what's throughout your respiratory system, except for a few areas of high trauma, like your epiglottis. It's going to have stratified squamous epithelium because it has to be a little tougher. And also, once you get to like respiratory zones and stuff, it kind of changes. It's not going to really have goblet cells because you don't need mucus there. Olfactory epithelium, right? So specifically, it's pseudostratified columnar epithelium without goblet cells. And it has these olfactory receptor cells that allows you to smell. The other two cells in the olfactory epithelium are supporting cells and basal cells. Respiratory epithelium is pseudostratified ciliated columnar epithelium with goblet cells. What does this mean? What does this look like? Well, pseudostratified looks like this, right? And the cilia are these little fingers, like they're little finger-like projections, okay? And they work to like push up the debris to the oropharynx to get it out, right? So these fingers are going and then pushing all these pathogens out. Columnar just means it's more rectangular shaped, column shaped. Goblet cells produce the mucus. Pseudostratified is, it looks like there's multiple layers, but there's really not. And you can think of the cilia as an escalator almost, or a filter. The mucus comes from the goblet cells, also submucosal glands, but it's highly viscous. So what does that mean? That's just referring to the thickness. So think like honey, right? Honey is super thick, super sticky. Okay, that's like the mucus here. And why is it? Well, it has to trap that debris and those pathogens, right? So it traps it in this thick mucus, and then it's sitting on top of the cilia, and the cilia kind of push it upwards. So think of like someone on an escalator, it's pushing them up. Now, it does sit on a hypophase layer, which is lower viscosity. So think more watery, more oily based, all right? And that just makes it easier for the cilia to move. So the mucus is on top, and then right here in between the cilia and stuff is that hypophase layer. Cells, goblet cells, they produce mucus. They also keep the membrane moist. You have brush cells, which are columnar cells with blunt apical microvilli, and they're chemosensory receptors that are part of the olfactory synapse. So anytime you see this, like blunt apical microvilli, chemosensory receptors, anything that has like a specific job, it's always good to take note of that because those are very testable. Specifically, when we get to this next one, the small granule cells, you'll see the diffuse neuroendocrine system with dense core granules. Anything that has dense core granules, you want to take note of that also because you can usually see that on imaging, which makes it very testable with images. And you're going to want to know what those granules look like. Now, basal cells, these are just stem cells. They're on the basement membrane. Nothing too exciting. Lamina propria. So this is deep to the respiratory epithelium, so underneath it, and it's made up of like very irregular, loose connective tissue, right? And that continues pretty much down till the gas exchange structure. So it's going like with that cilia and goblet cells, really. They have seromucous glands that provide moisture. They also have a large venous plexus system, so that in the nasal cavity. So that's kind of responsible for that stuffy feeling. When those get engorged or dilated, that can feel like stuffiness. They're responsible for warming the air though that's coming in. It also has lymphoid elements. Your larynx. Okay, so this is still part of the conducting system, right? Your larynx starts at the epiglottis and goes to the trachea. It has hyaline and elastic cartilage. Specifically, what has elastic cartilage is the epiglottis. The epiglottis is a really fun structure. I love it. It's kind of like a revolving door, or like a trap door. So basically, you have your epiglottis, uh, epiglottis that blocks like different pathways, right? So you have your trachea right here in front, and then you have your esophagus back there. Your esophagus leads to your stomach and your GI tract. So that's where food and fluid goes. But air has to go to your trachea. So the epiglottis is right there. So when you swallow something or you drink or eat something, it goes and traps the trachea. So you can't get anything through the trachea to the lungs because your lungs don't want that fluid. The lungs don't want food in it. And then it just allows that food and fluid to slide down to the esophagus. Now, when you breathe, okay, it flips and it goes and covers the esophagus. So nothing can come right here, right? But it allows that air to slide down the trachea into the lungs. So it's just blocking the different pathways, really. And I always think of it as a rotating door. It's made out of stratified squamous epithelium, right? So, like we said, high trauma areas, it's going to need that stratified squamous epithelium. This is a high trauma area. You have food, you have fluid hitting it, all kinds of things. So, it's all stratified squamous epithelium except for the inferior posterior part. That's where the respiratory epithelium begins. Now, bronchi. Okay, so the trachea, you come down, the trachea divides into two primary branches, your left and right primary bronchi, each leading to each lung. All right, there are 23 generations of branching, but the first branching is the secondary bronchi, sometimes referred to as lobar, and then the next one is tertiary or segmental bronchi. And then as they branch more and more and more, they get smaller and smaller. They have less smooth muscle, and they lose their C-shaped hyaline cartilage rings, right? So if you feel your trachea, this is your trachea right here in the front of your neck. If you feel it, you can actually feel those rings, right? That's that hyaline cartilage that they're referring to. Bronchioles. So your bronchi branch and branch and branch and branch and branch, and they're getting smaller every single time. When they get to about 1 millimeter, they're referred to as bronchioles. Okay. Now, the larger bronchioles, these are lined with simple ciliated columnar epithelium, and they have goblet cells still. Simple ciliated columnar epithelium, that just means it still has the cilia, and it's more rectangular shape. When you get to the smaller bronchioles, it starts to transition to simple cuboidal ciliated cells without goblet cells. So that just means it gets more square shaped, and it doesn't have any goblet cells, but it still has those little finger-like projections. Why? Why is it needed if it doesn't have any mucus? Well, it's just as a backup. In case that mucus happens to slide down to these smaller bronchioles, there's still cilia to push it back up. Why don't we want mucus down there? Well, when we get to the respiration zones, it has to be super thin for our gas exchange to occur. And if mucus is there, it's making that barrier super thick, right? So you have your alveoli up here and you have your capillaries down here where the blood is. And the gases have to get past this layer. Now, if you make it super thick and you just keep stacking on mucus layers and mucus layers, it's not going to be able to get through. The gas isn't going to be able to get through, and there's no gas exchange. So we have to keep that barrier really thin. Club cells. These were formerly called Clara cells, but they're dome-shaped cells with apical granules. So again, notice that granules, that's always big on imaging. These secrete lower viscosity, non-mucinous proteins and phospholipids, right? So this isn't like goblet cells. They're not secreting the honey-like fluid. They are more responsible for detoxifying harmful airborne substances. And they also contribute to the immune system by producing secretory components for IgA transport as well as lysozyme, antimicrobial enzymes, and cytokines that regulate local inflammation. And these are just all parts of the immune system. They also serve as progenitor cells for bronchiolar epithelial regeneration. Okay, respiratory bronchioles. If you remember the one of the first slides, this was the first part of the respiratory division, right? They have alveoli. It goes from alveolar ducts to alveolar sacs, and then alveoli, which are those grape clusters, right? And the alveolar ducts have simple squamous epithelium, right? Because simple squamous is very flat and it means just one layer, right? Now, these alveoli, these are tightly packed, right? And they even share walls called intra-alveolar septa. This really becomes important when you get to the pneumonia lectures because you have interstitial pneumonia, which can inflame those interstitial walls. They also contain small pores of Kohn that equalize air pressure and allow airflow between adjacent alveoli if a local airway is blocked. What does that mean? Well, you have all these alveoli, right? And say one gets blocked and collapses. You don't want all these others to start collapsing all over the place, right? You want to keep them open. So they have these pores. Type 1 pneumocytes. Okay, so now we're going to get into the alveolar cells. So they have different cells. They don't have goblet cells or anything. They have, this one is the majority. This makes up the majority of the surface. These are type 1 pneumocytes. They're very flat, very thin on imaging. Why? Well, we kind of already mentioned it. It has to be thin. That barrier between the alveoli and the capillary has to be thin so the gases can actually facilitate through. And that makes up type 1 pneumocytes are going to make up the majority of the surface. Type 2 pneumocytes or septal cells, these contain lamellar bodies, but their big thing is they secrete surfactant. All right, surfactant reduces surface tension and prevents collapse of the alveoli. So specifically, your alveoli, they're not all equally sized, right? Some are smaller, some are bigger, blah, 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 right? All the grapes are different sizes. Now, the smaller ones are going to have a lot more pressure. And if they have all this pressure, they're more likely to collapse, right? And we can't have alveoli collapsing because then we don't have air exchange. So we have surfactant being secreted by these type 2 pneumocytes, and the surfactant reduces the surface tension and kind of equalizes it throughout the alveoli. This is really important in preemies like babies born too early, because if they're born too early, their pneumocytes, their type 2 pneumocytes are not developed and are not secreting surfactant. So that's why sometimes babies are sent to the NICU because they need help breathing because they don't have this surfactant and they don't want the alveoli to collapse. But usually we're pretty good with type 2 pneumocytes, with medical advancements and everything. It's we're able to help them even earlier now. Alveolar phagocytes engulf debris, sometimes called dust cells. They're like macrophages. Alveoli to capillary. So we already kind of said it. We're talking about that barrier now, right? So here's the alveoli, here's the capillary. The capillary has the blood, the alveoli has the air. And we want to get the O2 from here into the capillaries and the CO2 from the capillaries into here so they can push it out. Now, this layer in between right here, all right, is three layers. You have the endothelial cells of the capillary, all right, so the capillary walls. You have the basal lamina that we talked about, and then you have the type 1 pneumocytes of the alveolar surface. And we're just saying type 1 pneumocytes because that makes up the majority of the surface. And that's the normal layer that it has to get through. And thank you for watching. That is the end of the introduction. Please stay tuned for our flu and pneumonia lectures, which will be coming up soon. Thank you.